And I hand over to my dear colleague and co-coordinator, Claire Corish, who will do the moderation of the second part.
Thank you very much, Dorothy, and thank you all. So it's my name is Claire Corish, as Dorothy said. I'm Professor of Clinical Nutrition and Dietetics in University College Dublin, and I'm very pleased now to welcome Dr. Dominique Dardevet to answer some of the questions I think that came up in the last session. And Dominique has a PhD in endocrinology, nutrition and metabolism. He's a full professor and deputy director of the Human Nutrition Unit in Clermont-Ferrand in France, in the National Research Institute for Agriculture, Food and the Environment. And he works in the Department of Human Nutrition. So with that, thank you very much, Dominique, for your presentation.
Yeah. So good morning to everybody, and I would like to thank the coordinator for including me in this very nice project. So we are going to switch to the more in vivo studies and the clinical studies according to the different PPF, which has been built by Stephanie at the Fraunhofer. Oops. Oh, it's not working. Okay, as you know, so with age, and it's well established now that the tissues and organs are less sensitive to the anabolic effect of dietary protein. And this is especially true for the skeletal muscle. This implies that the requirements of protein has been increased for the older population, even if they are considered as healthy. And so in some countries and European countries, the recommendation of protein intake has been increased by 30% from 0.8 grams per kilograms and per day to 1.2 grams of proteins per kilograms of body weight and per day. However, this recommendation is based on good quality proteins. What is a good quality proteins for older adults? This is a protein which generates high plasma amino acid concentration after ingestions, which has no limiting amino acid, and which has a fast digestion speed. So in other words, at same amount consumed, it's better to have a plasma amino acid profile like the orange protein than the purple protein. For the older adults, there is some other consideration to take into account. So some amino acids, such as leucine, has to be increased in the protein intake. We need to have about 25 to 30 grams of protein in a single meal. And we have also to limit the matrix effect, the nutrient interactions, and the effect of processes, which could affect the digestibility, which could decrease the digestion speed of the dietary protein, and which could also decrease or we may have some amino acid loss in all proteins. So when we are in the transition to incorporate more vegetable proteins in compensation to a decrease of animal proteins, so we need to take into consideration all of these parameters. So we know that the plant proteins are of less quality than the animal proteins because they have some limiting amino acid. So this has been mentioned by Stephanie before. So compared to the FAO proteins, the pulse or legumes proteins have the sulfur amino acid as limiting amino acids, and for the cereal protein, this is the lysine. Or whether, as mentioned by Stephanie, it's not a problem if we are mixing adequately different sources of plant proteins. So this is just a case here where we mixed the pea and the wheat protein in such amount that we have no more limiting amino acids when compared to the reference protein. Or whether because the lysine is a very high limiting amino acid in cereal, so we need to have blend of vegetable proteins with between 70 and 80% of pulse of legume proteins compared to the cereal proteins. So as mentioned by Stephanie also before, so we tested three ppf according to their acceptability and taste, developed by Stephanie. So this is the ppf3, ppf5, and ppf6. And here you have the different isolate or concentrate of protein used in this blend. So the 3 ppf has been selected from their acceptability by the target population. And so we included this ppf in an experimental meal in order to measure the postprandial amino acid kinetics and then the apparent bioavailability of each amino acids. So we calculate the amount of ppf protein in the meal in order to provide the same amount of leucine than 30 grams of whey. The whey is considered as the most optimal animal proteins to the age population when given at this amount. So we calculated the real amount of each amino acid ingested by a direct amino acid assessment in the meal, and we measured the amino acid levels in arterial blood every 30 minutes for 3 hours. And we calculated the incremental increase of each amino acid above the basal value. So we tested this on 11 volunteers from Ireland and Italy. The mean age was 74. The control meal contains 30 grams of whey, and the ppf meal are adjusted in order to have the same leucine intake. So this is the composition of the meal given to each volunteers. So they are equals in carbohydrates, lipids, and in order to have the same amount of leucine, you can see that we had to increase the amount of proteins from the ppf compared to the whey. So we measure directly in the meal the amino acid composition. So as you can see here, so we have the same leucine intake between the wheys and the ppf proteins, and we have the same amount of essential amino acid ingested. For some amino acids, we have even more some amino acids in the ppf than in the whey. This is the case for arginine, tyrosine, methionine, and phenylalanine. But in some cases, or for some amino acids, we have lower amino acid ingested than in the whey, and this is the case for the threonine, cysteine, and isoleucine. So just to be sure that we had no limiting amino acid into the meal, so we compare the amino acid ingested with 30 grams of whey or 30 grams of the FAO proteins compared to the ppf ingested, which was 40 grams. And as you can see here, compared to the FAO, we have all the amino acids ingested and provided by the ppf were at least equals or superior to the ingestions of 30 grams of the FAO protein. So we have no limiting amino acids in all this. So what is the amino acid kinetics now? So as you can see here, for the whey in blue and for the pre-ppf here in yellow, gray, or orange, you can see that the digestion speed of the ppf is lower than for the whey by about 30%. And this is a direct consequence. So we have a lower peak of amino acidemia, which is decreased by 30% with the ppf compared to the whey. So if we calculate the apparent bioavailability of essential amino acids, so we have a slight decrease, which is almost significant for all the ppf. It's a decrease by about 25%, despite that the same amount of essential amino acid has been ingested. For the leucine, we have also a decrease of 25%, even if this decrease is not significantly different between the whey and the ppf. However, if we look at the maximal amino acid concentration after ppf or the whey proteins, we have a decrease of the maximal amino acids concentration with the ppf. However, so we see that for some amino acids, we have a little bit of a problem, because if you look at the threonine, for examples, so we have a significant decrease of the apparent bioavailability of threonine by 60% in the ppf compared to the whey proteins. but we have only a 30% decrease of ingestion. So this is not related, and so we have a specific effect of the PPF on the threonine bioavailability. And so we recorded the same consequence with the sulfur amino acids, with the cysteine and the methionine. In the cysteine, we have a lower intake of cysteine in the PPF, but we have no increase of cysteine to the plasma after the PPF injections. And with the methionine, despite a higher increase or ingestions of methionine, we have no increase of methionine into the blood after the PPF consumption. So it seems that with the plant protein sources, the apparent peripheral bioavailability of these three amino acids is seriously decreased in older adults. So the conclusion of these first results is that the plant proteins we use have a lower digestion speed than the whey, and this is resulting in a lower maximal concentration of amino acid after the intake for the leucine and for the essential amino acids. So for plant proteins and for some amino acids, there is a higher decrease in their arterial bioavailability, which seems to be independent or not totally dependent on their intake. And this is the case for the threonine, the methionine, and the cysteine in our studies. So what could be the explanation? So do we have a lower digestibility or the presence of anti-nutritional factors in the PPF protein sources? This is unlikely because this would have affect all essential amino acid kinetics. So maybe we have the second explanation is that we could have a higher splanchnic extraction of some amino acid with plant protein sources than with animal protein. And this is a more likely explanation. What is the splanchnic extraction of amino acids? So after ingestions of proteins, so we have a protein digestion into the gut, and we have amino acid absorption into the portal vein. It's reaching the liver, and then it's distributed to the peripheral tissues, including muscles or other organs. So some amino acids are taken by the intestine or by the liver, and this is what we call the splanchnic extraction. And of course, the higher splanchnic extraction is higher the decrease of peripheral amino acid bioavailability is. So we also know from the literature that the splanchnic extraction of amino acid is higher with plant protein than with animal protein, and we do not have any explanation yet, of this difference. And we also know that the splanchnic extraction of amino acid is higher in older adults than in adults. So, of course, the question is splanchnic extraction challenging the anabolic effect of plant-based dietary proteins in older adults? And this is what we are actually checking on ongoing clinical studies in which we are going to compare the whey and the plant protein on the single volunteers to measure the amino acid bioavailability and splanchnic extraction of amino acid from these two sources and the whole body and muscle protein anabolism. And we are going to implement 12 weeks of adaptive physical activity programs, and we measure these parameters from whey and plant proteins before and after the adaptive physical activity programs in older adults. So why physical activity chosen? Because we know that the physical activity decreases inflammation. It could decrease the inflammation at the gut level, and this will decrease the amino acid requirements in the splanchnic bed and increase the amino acid bioavailability for the other tissues. And it also decreasing the inflammation at the systemic level. And we know that the inflammation or the systemic inflammation is responsible for the anabolic resistance of dietary protein intake observed in older adults. And also because the physical activity decreases fat mass and increase insulin sensitivity. And at the tissue level, an increase of insulin sensitivity could increase the utilization of dietary amino acid for protein synthesis and anabolisms. So this is all for me today. And if you want to know more about these studies, we published these results. And I thank you for your attention and waiting to take any questions.
Thank you very much, Dominique. And there are a few questions already in the Q&A. The first one is, did you analyze the different amino acid content in single combinations, or is it based on theoretical data, based on a nutritional database?
So we analyzed, the different amino acid content. So we measure in the meal directly the amino acid composition, because of course we had to build the meals, the theoretical or the manufacturer information, but we really wanted to be sure exactly which of each amino acid were ingested in order to compare the meal, and just to be sure that we are no limiting amino acid, and just to compare the each amino acid bioavailability compared to their real ingestions.
Thank you. And there's another question here now from Anna-Marie Beck. And I think it helped, the work that you did helped inform our approach to work package 4. But I suppose Anna-Marie is just asking, I'm not quite sure I understand the problem here, as long as older adults are not vegan. So what would you like to comment on that, Dominique?
Okay, it's true that so far, in the general population in Europe, there is a very little vegan for the adults. However, as mentioned before, the people usually decrease their protein intake. So they are, most of them, or they are tend to be the lower the recommendation, which is 1 grams of proteins per kilograms and per day. Some are at 0.8 and even lower. And of course, we know that this is not covering the amino acid recommendation for these other requirements for these older adults. Of course, if we are taking, or if you are increasing the protein intake of vegetable proteins and not increasing your protein intake, then you could have some limiting amino acids. And if you have a decrease of amino acid bioavailability, even if you are at a lower recommendation, which is 0.8, and if you have a lot of vegetable protein, then you will be under the recommendation and you won't have an optimal anabolism for this population. So this includes also the people we are not vegan, and we could also maintain a part of animal protein in their diet. So of course, if these people are eating more than 1.4 grams of protein per day, this won't have a major consequence on their anabolism. But here we are targeting the people with lower appetite, and we are just at the recommendation of Vito.
Okay, and then one final question before we move on, Dominique. Given your results and considering that hospitalized older adults are typically inactive, undernourished, and already consume insufficient amounts of protein, would you currently recommend increasing plant protein intake in this population? Or I suppose, what is your comment on that, on the evidence for this?
Yeah, of course. We didn't have the result yet of our clinical study to compare the anabolic effect of the PPF compared to the whey. Of course, but we assume that we will have a difference before the physical activity program. So probably the plant protein will be of lower anabolic effect than the whey, even if we are increasing the amount to 40 grams compared to the 30 grams. Especially if you are just at recommendation. Of course, the physical activity, we expect, but maybe we will be wrong, that by decreasing inflammation and increasing the tissue sensitivity to amino acids, we may recover, or the plant protein will be of a higher anabolic effect. Of course, in hospitalized patients, first because they are not eating a lot, and because it might be complicated to have an adapted a physical activity program in such population, it might be difficult to implement. Or whether by trying to maintain these people in a minimal physical activity could be helpful. If you want to increase the potential or the anabolic effect of protein intake in such population. Of course, if you can do that, if you want to switch for more plant proteins in the diet, probably to counteract the lower quality of plant protein, you will have to increase more the plant protein than the animal proteins. So you may assume that if you want to be at 1.1 grams of proteins per kg and per day, with the regular protein, if you want to increase the plant protein, probably we have to go for 1.2, 1.3 grams of only vegetable proteins in the diet to be sure that you will cover the requirements of such population, and that might be difficult to achieve in such population with lower appetite.
All right, thank you very much, Dominique, for that important work, which really informed, together with Work Package 1, the approach to Work Package 4. So if I move on now if you stop sharing and let Giuseppe start to share, and I'll introduce Giuseppe. There are a couple of other questions, but we just have to maybe answer them at the end. And if Giuseppe can share now, and I will introduce Giuseppe. All right, so again, it is very much my pleasure to introduce Professor Giuseppe Vito. Giuseppe is a medical doctor with a PhD. His background is both in medicine, surgery, and sports medicine. He's currently full professor of physiology in the University of Padua and working in the Department of Biomedical Sciences and the medical school. And I'll hand over now to Giuseppe to present some of the work that they did in Padua on appetite. Thank you, Giuseppe. Giuseppe, are you okay? Are you muted? We can't hear you.
I want to say thank you very much for this nice introduction. In Padua, obviously, we participated in two work packages. The work package 3 that has been already presented partially by Dominique just in the previous presentation, and also in the work package 4. So, but at the same time, we were interested in investigating some aspect of the neuromuscular functioning in the older. So we added the specific test in our project, and namely we added the ultrasound imaging to assess muscle architecture and volumes. And then we also collected from some of the volunteers, not all volunteers accepted to receive a muscle biopsy, to assess fiber typing, cross-sectional area, macro denervation and muscle signaling. And also I place this in reds because these are the two main aspects that I'm going to cover today with my presentation. We were interested in a blood-based biomarker of neural degeneration that nowadays are considered one of the major factors for muscle dysfunction in aging. And also we use a technique to assess in vivo motor unit behavior, that is the high-density electromyography. Okay, if I move this slide. Okay, it's well known that with age we tend to have a reduction in all physiological function. In particular, the muscle mass is reduced, and this is accompanied by reduction in muscle strength. But it's also well known that the muscle strength is running more rapidly. The muscle strength decline is running more rapidly than the muscle mass loss. So when you normalize the muscle strength to the muscle volume, muscle mass, you see that there is still a difference between old and young people, as simplified in this figure here. It's a study done already more than 20 years ago. And at the same time, there is a growing evidence that the nervous system contributes significantly to the deterioration in neuromuscular function. And this has been demonstrated, for instance, indicating that older adults, especially the frail adults, present a reduced ability to activate fully their muscle. At the same time, there is a reduced corticospinal excitability that's been measured. And also the spinal reflex excitability is reduced, as simplified here by the experiments which use the H reflex as a marker of spinal excitability. Obviously, this is also sustained by a loss of motor units that are the unit that are regulating muscle contraction and their remodeling. Among this factor, one that is gaining more attention is concerning the neuromuscular junction. Neuromuscular junction, the chemical synapses linking the nervous system to the muscle, is now recognized one of the key factors in the deterioration of the muscle with aging, and especially in sarcopenia. This image here is an image taken from our lab from muscle biopsy, we are able in some case to identify and to have a morphology of the neuromuscular junction. But obviously this kind of analysis is not easy to do. So we need to also to look at markers that are more easily to assess in the clinical set. So this why we focus also on biomarkers that are blood-based. They can be easily sampled from blood samples. In particular, we focus on two blood-based biomarkers, the CAF, also known as a C-terminal agrin fragment, and the BDNF. Today, I will focus mostly on the CAF. What is the CAF? CAF is a fragment that is detached from agrin. Agrin is a crucial protein which maintain the integrity of the neuromuscular junction, orchestrating the right folding of the presynaptic and postsynaptic area. So when there is a damage, when the subject for instance, is in bed or during the disuse, the agrin is fragmented, and in the blood, the sick fragment appears. And then you can see that in muscle wasting disease, this is higher. And also, we demonstrate in a very healthy young people that this can be elevated also in people that are in bed and are young and healthy. So it's a good marker, very sensitive. And also, we recently published reference values for this parameter. So at the same time, we wanted, as I said, to investigate the motor unit behavior in vivo, because we adopt a technique that is based on high density electromyography, which an array with the multiple electrodes attached to the muscle. In this case, we use the vasus lateralis, and we ask the subject to perform different isometric contraction, triangular or trapezoidal. These contractions are progressively growing and decreasing, and this way we can look at the motor units that are progressively recruited. We have a very open window of the behavior of the motor unit in vivo, because we can identify, for instance, the point of recruitment on the curve. So we can identify at which level of force the motor unit is recruited. In this case, this one is recruited here. And at which time this motor unit is de-recruited. This is a very important information. So at the same time, also, we have the force that is maintained for a period, that we can assess other parameters such as mass steadiness, that is the capacity to maintain the force output constant. It's a critical factor, for instance, in aging. And finally, another parameter we consider was the persistent inward current, that is the level of neuromodulation. Neuromodulation is a very important parameter, factors which contribute to the control of motor unit. When a motor unit is activated, the level of serotonin and norepinephrine, two monoamines, dictate how fast the motor unit is firing, and they can also prolong the firing rate. The neuromodulation is decreased with age, is decreased with disuse. It can be improved by an healthy situation, and also can be increased by training. Now, how we approach this analysis for representation? We decided to have a dichotomic approach using CAF, the level of C fragment, and the parameter we measure in blood. We divided the group into two groups. The one using median split, we have low CAF people and high CAF people. I remind you that the low CAF people are those that are in better shape, because high CAF people are those that are exhibiting neuromuscular degeneration. And you see here the results. You have the high CAF and low CAF group. And then we can say that in spite of this huge difference, the maximal strength in this group was not distinct. So this was not affected, although the variability in the high CAF group is much larger. Also, other characteristics, we found that in the two groups, the physical characteristics, body composition of BMI, body Randex index, nutritional profile were not different between the two groups. Just a small difference, not significant, was in terms of physical activity, with the low CAF individuals exhibiting a slightly higher level of physical activity. And now, what are the results using this analysis? We found that as expected, the persistent inward current, the neuromodulation, what I just mentioned, is higher in the people with the low CAF concentration. But contrary to our expectation, the steadiness, the capacity to maintain force, instead is better in the people with the high CAF. So this is something we don't know how to explain. We are trying now to look at mechanism potentially underlying this kind of odd result. But we want also to relate this to the appetite, because, just not to be completely out of the context. So we try to correlate this figure with appetite. We use a number of correlations. We found that the snack score correlate with the HOMA, an indicator of insulin sensitive insulin resistance, correlate negatively with the HOMA. So people with a high appetite tend to have a low HOMA score. At the same time, the same results applies to insulin level, which is reduced in people with a high snack score. And interestingly, also a functional test that is also a measure of force, the strength, the 5 sit to stand chair test was also associated with the inversely with the snack score. So indicated people with a better snack score, better appetite tend to perform better in this test. I remind you that these people were not distinct in terms of mass strength, the mass strength, but this parameter is correlated with appetite. So, and I forgot to say that here you can find the two groups together. The green dot are referring to the high CAF and the blue dot to the low CAF. So in conclusion, although our preliminary results did not show any change in terms of neuromuscular function across the groups and the intervention when we are looking at the work package 4, but work package 4 is a big study, with a lot of participants. Our group was smaller, so the statistical power our group is definitely too low. But when we consider the CAF, that to us is a very good biomarker, easy to be measured, we can say that in spite of no difference in BDNF, the BDNF was not different in these two groups of using CAF. The two groups exhibited similar mass strength, similar body nutritional body composition, similar nutritional intake values, and in general they were in quite good health. But the motor unit analysis revealed that the neuromodulation and the discharge rate, especially at the recruitment of the motor unit, was higher in the low CAF group. So the people that are in a better shape in terms of neuromuscular junction performance. But contrary to our expectation, muscle force steadiness was higher in the people with the sign of the degeneration of neuromuscular junction. This is something we wanted to investigate further. The snack test correlate negatively with the insulin concentration, the HOMA, and with the 5-time chair raise testing, with no differences observed between the two considered groups. And this was something preliminary. We are now exploring more in depth all the other information we collected in this study. But before I conclude, it's important I recognize the contribution of all the people that participated to this study, and they made this possible. Thank you very much for your attention.
Thank you very much, Giuseppe. Are there any questions? I don't see any questions yet in the Q&A. So maybe I will just start by asking Giuseppe. Giuseppe, you said you were going to do some more analysis that's related to work package 4 and work package 3 and work package 4. Maybe you'd like to just explain what the plans are for that and what you think are the important things to focus on.
I think as I show, we measure a lot of data, extra data. We wanted to have a more complete assessment, including all the other parameters. For instance, we did On the people who accepted to be biopsied before and after the intervention, we did some preliminary analysis. As the first analysis did not show any significant change, but I think there is the opportunity maybe to amplify this analysis. For instance, we were looking at the denervation in the muscle. The initial early investigation did not produce any difference in the people that were biopsied before and after. But unfortunately, we had 24 people who underwent to a biopsy procedure in the first at the baseline, but only 18 accepted to be biopsied after the intervention. Therefore, this is undermining our statistical capability. Is the
Yes, I
and
suppose
then, you know,
muscle biopsies are probably not everybody's lining up to have done. But so well
I
done,
know,
really.
I don't know, maybe
Yeah,
Irish people were more
acceptable.
courageous in terms of the acceptable.
All right. Okay, Giuseppe, as there's no more questions in the, I mean, if people want to ask questions later on, I'm sure Giuseppe will answer them. But I think then we'll just move on. I'll hand back then over to Dorothy to pass over to the next moderator. Thank you, Giuseppe, very much.